Okay, so welcome back. As always, any questions about stuff I've covered or any follow-up questions for Mike or anyone else? I think it's always good to set that. And again, I want to keep on reminding people that, take notes, and if you have questions and don't want to ask them during the main discussion, certainly catch us, because I'll be around all 2 weeks and Mike will be around through Wednesday, and I think Nick is around most of the time as well. So most will be around for a long period of time. And so please feel free to catch us at lunch or breakfast or dinner. And like I said, if there's an open seat at our tables, please sit down, right? Don't be bashful about that. So what I want to talk is last time I talked about the way we thought originally about trying to find underlying segregating variants. And that's what was called linkage mapping. And the idea there is we simply look for an association in the gametes from a parent or in a pedigree between a marker and the trait value. So we look for marker trait associations. And that was conceptually simple to do. The problem was it's not a very powerful design because you need to have a single family. And for most species that limits you to several hundred. Even a well-designed plant breeding experiment, maybe you've got 1,000 or so. And also, those chunks of chromosomes you carry along that are unrecombined, they're tagged by markers, are quite large. So now I want to talk about the way that we do it now, which is association analysis. And the key thing that makes association analysis work is that with a linkage-based QTL study, you can get 50 to a couple hundred markers and be fine. A couple of markers per chromosome. To do GWAS, you need tens of thousands, if not millions of markers. The flip side is now your sample isn't a single family, it's an entire population. So you can have GWASs which are in the hundreds or 200s or 300,000s. You can also apply this technology, this methodology to much smaller sample sizes. So GWAS. Here's the overview. I'm going to talk about GWAS, give a quick overview of what the results say, and I'll flesh those out a bit later. Then a lot of it depends upon LD, and there's several different measures of LD. In fact, there's this distinction between complete disequilibrium and perfect disequilibrium. And it turns out that's really important because a very subtle feature that drives a lot of our analysis that we'll talk about is we use markers, and the markers we typically use are chosen because they're polymorphic. Therefore, they're what are often called common markers. The minor allele frequency is 5% or more. It turns out that if you have a causative site and a nearby marker, even if you're in LD, if the allele frequency mismatches, so the minor allele frequency here is at 1%, the minor allele frequency at your marker is 20%, that mismatch means most of the LD isn't captured by it. So a lot of the early GWASs used common markers and didn't capture a certain fraction of variation. That's, for example, where missing heritability comes from. So we'll talk a fair bit about that. It's a little technical, but I'll highlight what the key points are. And the main thing is simply this, and this, by the way, comes up again and again and again in genomics, is that you have a site frequency spectrum for markers. Then you have a site frequency spectrum for causal alleles, or if you're doing inbreeding depression, for alleles involving inbreeding depression, namely those loci that show directional dominance. If you have a mismatch between the site frequency spectrum of markers and the site frequency spectrum of your causative sites, your analysis isn't biased, but it's much, much less powerful, and you end up missing a lot of stuff. Then we'll talk about this really exciting development that's happened in the last 10 years or so. We typically think about a quantitative trait as height, or weight, or cognitive fashions. But anything we can measure is a quantitative trait. So for example, I could measure whether or not a specific promoter is activated or not, whether there's a factor boundary, whether the histones are open. I can measure the level of gene expression. That's a measurable trait, and therefore I can apply quantitative genetics. I can look for QTLs or SNPs that influence expression level. Some of those are right nearby, not unexpectedly. Some of those are on different chromosomes. So we now have this new field I call quantitative genomics, what we're measuring is not just simply a trait like height, but we're measuring things which are upstream from that trait, like expression level, like protein amount, like splicing isoform ratios, like polyadenylation status. And so all of these molecular intermediates we can treat as quantitative characters, and those are often used to try to get a bit more inference. So we'll talk a little bit about that. It first comes up when we talk about eSNPs, then we'll talk more generally about reg-SNPs, so we'll talk about cis-trans, then we'll talk about this idea about mediation, and then we'll close up by talking about what do we know about GWAS, what does that tell us about architectures, what does that tell us about selection. So that's kind of the journey we'll be on. Hopefully we won't get too lost. As always, there's a lot more that I'm going to cover, and you'll see in some places I've been very lazy. You'll see a nice cut and paste. That cut and paste is right from one of these two chapters, so you can look that up. So chapter 20 covers GWAS, 21 covers quantitative genetics, and mediation analysis, which involves path analysis and also something called Mendelian randomization, is discussed in Appendix 2. I'll make sure all those are posted. So the idea about association mapping is really simple. If when I sample chromosomes, that markers which are really close to each other, by really close I mean maybe 10 kilobases or less, if markers that are really close to each other tend to be correlated, so if I get marker big M, I tend to get things nearby on that same chromosome, that's called LD, and that then creates a marker trait association. So just like in QTL mapping, where I could take a marker and then take individuals and put them into marker homozygote 1, marker heterozygote, marker homozygote 2, and ask if there was a trait difference, that is, look for a genotype trait association or a marker trait association. I use the exact same methodology, except now what I'm sampling is not a family, but a large random population. And almost all markers aren't going to show this, but there's a tiny subset of markers that are going to be very close to causative sites that will show this. This is why you need tens of thousands, or more likely hundreds of thousands of markers, and we'll all flesh that out. So this involves population level LD. The downside is a huge number of markers. This has only become feasible with modern genomics and chips and whole genome sequencing. The good news is that when you map a site, that marker that's tagging a region is not tagging a region that's 20 to 40 megabases. It's now tagging a region, depending upon your species. It could be under a kilobase, for example, in maize, or it could be 20 or 30 kilobases in humans. So it's now tagging a much, much smaller region. So it's like you can think about GWAS saying the globe is my genome. And GWAS says, hey, we know you're somewhere in South Central India. That's what QTL does. That's good. It's not a street address. GWAS says you're in the city of Bangalore. Again, you're a lot closer, but you're not right on the spot. So these are levels of getting closer and closer resolution. So here is how we generate LD. It's really, really simple. Let's imagine I've got a chromosome, and it's got a SNP on it. So I've got a big M and a little m, and these are neutral SNPs, and I can simply score them. And let's imagine that in this population, 45% of all chromosomes are big M, little q, where q is the allele that impacts the trait, or locus that impacts the trait. 55% are little m, little q. Now let's imagine that somewhere back in time, little q has mutated on the M background to big Q. So now 1% of all chromosomes are this big M. I thought there was a cockroach on my neck or something. I could feel something moving back there. We'll get this sorted. Hang on a second here. There, how's that? Sound good? Sound quality good back there? Yes? Can you hear me? Is this yes or this no? I'm seeing kind of a, so if you can hear me fine, raise your hand. Okay, good, good. So now I've had little q on the M background mutate to big Q. So now here are the frequencies of chromosomes, the population, and now what you see is now if I have big M, there's some chance I have a big Q. If I have little m, there's no chance. So that if you carry a big M, then on average, you might be just slightly heavier, slightly larger, or so slightly more levels of transcript. So it's a small signal. But a modern GWAS has a sample size of several 100,000 individuals at a minimum, so you have incredible power. The flip side is you also, really tiny ascertainment errors can translate into big effects. You've got to really be careful because GWASs are so powerful, little sources of bias can really influence, like population structure, and we'll talk about this. So this is mutation, which is now generated LD between our marker and a SNP now that impacts our QTL. And the idea, just like in a QTL study, is I have now 1 million SNPs. I go to SNP1 and I say, break individuals into 3 genotypes based upon SNP1. Is there a difference? No. SNP2, no, and move along. And that difference I could either do an ANOVA and look for differences between a continuous trait, or I could do chi-square tables, or logistic regression, or Cockrum-Armitage trend tests if I have discrete data. So all that methodology we use for QTL mapping for marker trait associations is exactly the same we use to detect them in a GWAS. And again, just to remind you what LD is, and here's the punchline down here, we're going to use this correlation between the frequencies. We'll spin that out in a second because it turns out that if I have a variation at this site, only R squared of that variation is passed on to the marker. So the marker variance is only R squared of the actual causative variance. So we'll talk about where that comes from. And so what that means, if I have some initial correlation, some R squared between M and Q, after T generations, this is how much remains. And so what you see is that this decays away for a long time unless C is really, 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 really tiny. And you can score that by simply taking your organism and looking at markers, looking at how long the average LD block is. And it ranges from under a kilobase to several hundred kilobases, depending upon your species. And I'll talk about that a little bit. So the idea is, conceptually, it's very easy. Get a really big sample, score traits on those big samples, score markers, and on each marker do an ANOVA. Hey, pretty simple, right? Well, there's some complications, but that's the basic idea. So conceptually, it's pretty easy to carry out. Scoring a lot of markers used to be a big deal. Now the bottleneck is phenotyping. It's easy to sequence your genome. Phenotyping, that's another issue, which is kind of an interesting irony. So the conceptual part about gathering the data and testing the SNPs is actually fairly straightforward. And then, of course, as we'll talk about, complications come in. So here's an example. This is from Ed Buckler and Mike Gore. So here we have LD ranges in the major crops. There's a reason I'm bringing this up, is if you look at, let's say, maize. So in wild lines of maize, remember maize in natural populations is an open pollinated outbreeding variety. The LD range is under a kilobase. Look at landrace under a kilobase. Look in elite inbreds, it's about 100 kilobases. Same thing over here. Sorghum land races are 5 to 50 kilobases. Again, this is an average size. It shrinks and expands depending upon recombination rate and past history. But for example, over here, if I'm, let's say, looking at land races in barley, I'd want to have a couple of markers, so I've got a couple of markers per 100 kilobases. If I've got more markers, I probably can't use that information. If I have less markers, I'm missing those blocks. Now, what accounts for these differences in block sizes? What did we talk about was the key thing to think about when thinking about stuff in population genetics? Time, right? Time back to common ancestor. If I take 2 individuals and their time back to common ancestor is long, then that coalescent path has had a long time for recombination to occur, and only really tight regions don't have LD broken up. Remember, 1 minus C to the T. If that T is short, then I have longer blocks of LD because I haven't had enough time to break things up. So, for example, let's look at maize over here. Land races are simply wild populations. They have very large effective population sizes, long time back to common ancestor. Elite maize inbred lines are recent developments and trace back very quickly to a really recent common ancestor. So your LD pattern is something you can score fairly quickly. And the idea is you want at least several markers per average LD block size. And in reality, people just kind of use everything because it's usually quicker to score things than to do that. So the idea is really straightforward. Hundreds of markers per centimorgan, basically luck for LD decay. The fraction, so if this term over here, if I have an additive QTL, this is the causal site variance. If R squared is the correlation between my causal allele and my marker allele, we'll talk about that a little bit more in a second, then the fraction of variation the marker accounts for is only R squared of the causal variance. So for example, if R squared is 10%, then this marker is only going to capture, it'll capture variation, but it'll only pick up 10% of the actual causative variation from the nearby site. Widely used since the early 2000s, especially as markers became more common. It's the mainstay of human genetics, it's widely used in breeding, and it's also making progress in evolutionary genetics. Now, human studies have massive, massive, massive population sizes. Those are nice, but you don't need them. As population sizes get smaller, obviously you have real power issues, but you can do a GWAS in an evolutionary or an ecological setting. You certainly want, ideally, a couple of thousand individuals, but if you get the right type of replication, you can even have smaller ones. So I've seen a nice GWAS that was done in 100 highly inbred Arabidopsis lines. And the nice thing about being highly inbred is in each line you can measure the trait with arbitrary precision. And that's an advantage. So if you can make inbred lines and replicate, you can do a GWAS with a much smaller sample size because you have a much more accurate measurement of your trait value. So the way we typically plot these is on something called, I'll get back to that in a second, something called the Manhattan plot. We've seen these before. So each pixel represents a test, and the chromosomes are color-coded here. And on this diagram here, come back to that in a second. On this diagram here, we basically plot minus either natural log or log base 10 of the p-value. So each pixel, and basically what I do is I scan across a chromosome, and each marker I test, I note its location, then I put minus log 10, that's my pixel up there. And so we set these thresholds by Bonferroni style corrections. I'll talk a lot more about multiple comparison and corrections, which are how you set those. I'll talk a lot more about that, I think, on next Thursday. There's a very robust debate in the human genetics literature, and chapter 20 has got about a page and a half of that as to what the value people choose is. And the value people typically choose is this thing about 5 times 10 to the minus 8. And again, the reason for that, there's some delicate issues in here. One delicate issue is there's a thing called imputation. Imputation means if I have a reference population that I've sequenced and I have a sample from that population, I've only scored using a chip. So a chip will score 1 million SNPs. I can impute or guess at things I haven't scored by looking at the haplotypes I've had scored and project them back onto the reference population. So the problem with that idea is if I set this by testing 1 million SNPs and I find certain ones are significant, I can now go back and take the exact same data and now impute it to 2 million SNPs. Now Bonferroni gets even smaller, and some things which were significant before I imputed are no longer significant. And so how people handle that is they kind of set a preset level beforehand and their Bayesian methods. It gets a little bit involved, but chapter 20, if you're interested, gives kind of a deep dive into that. And I can talk about that afterwards, people, if they want that as well. So it's called the Manhattan plot because it looks like the skyline of Manhattan, right? Now, people get whimsical. If you have a really, really good one, we call that a Dubai plot, right? And if you have a really, really bad one that is it's flat, we call that a Manhattan, Kansas plot, right? And so the idea is as your sample size gets larger, you can move from Manhattan, Kansas to Dubai. And people have things like they call a Manhattan sunset plot when they use color coding. So there's a lot of colorful terms in literature. So if you look at it here, that's solid not because there's no information there. It's because there's hundreds of thousands of pixels in here. Anybody know why there are these clear spots here? Some are clear between chromosomes, but some are in the middle of chromosomes. Those are centromeres. Around centromeres, there's often very reduced levels of variation. And so on a lot of plots, you often see these gaps in here. Sometimes you see them between chromosomes, but those are often because they're acrocentric and the centromeres are on the end. So let's imagine I see this. Look at that. Wow. Pretty. We'll come back to FTO, by the way, in a second. That's actually interesting. So here I have this, and why do I get this massive spike up there? Well, you get that massive spike. If we blow up that region, we look at something like this. So to give you some perspective, this here, that's basically about 600 kb bases. So now when I blow up that, they see each individual pixel then resolves. Then I have the lead or sentinel SNP, the SNP with the highest value. Then I've color-coded how they are with that. This also has localized recombination rates. So when you blow up one of these regions, you get a lot more structure. So if I took this and just spread it apart, those would become individual pixels, and these peaks would still be a peak, but they'd spread out a little bit. So misconception number one, the lead or highest SNP is the SNP closest to the causal site. Wrong. We'll cover that. You go, well, that's okay. I've done whole genome sequence. I've actually sequenced the causal SNP. I don't know this, but I've sequenced it. Therefore, the causal SNP has to be the highest scoring SNP, right? Wrong. We'll talk about some of those things and how you can combine them. So for common alleles, there's not much to be gained from moving to high density SNP to whole genome sequencing. For rare alleles, there is much to be gained. Again, we'll flesh all that out. So far it seems pretty straightforward. Get a big sample, look at marker trait associations, do multiple comparison corrections. The problem is a modern GWAS, because its sample size is so large, has incredible power. And as Spider-Man said, or Spider-Man's uncle said, with great power comes great responsibility. And so GWASs have enormous power, and so subtle features can give you massive signals. And one subtle feature people were aware early on is that association does not imply linkage. And the argument is actually very straightforward. Suppose I have two populations that I don't know that I have two populations in my sample. And one population has a high disease risk, and one population has a low disease risk. Likewise, populations differ in that I have some alleles which are much more common in one population than the other population. So an allele that's more common in the high disease population will show a higher disease risk, not because the allele is in linkage with the causal site, but rather that allele predicts group membership predicts trait. So this is the problem of population structure, and it's one that we have to worry about getting around. And here's the early paper before GWAS that got people really seriously thinking about this. So I live in southern Arizona in the US, that's just east of California and right on the Mexican border. And there's several Native American nations around there. And they have, especially the Pima, have a very high risk of adult diabetes on a Western diet. So there are a lot of studies been there because of health reasons. And so GM is a marker that's in a metabolic pathway that was thought to be an obvious candidate. So what they did was they went in and they looked at it and they basically scored and found that about 290 individuals had that marker and about 4,600 didn't have that marker. And if you didn't have that marker, your diabetes risk was a lot lower. You do not need statistics to tell you that there's a significant difference, right? I mean, I think chi-square is probably like 10 to the fourth or something. So there's a massive difference. So you might think, I'm going to go to a big drug company and get this because it's obviously a gene that lowers risk for diabetes. Until people said, wait a minute now, the GM haplotype is rare in full heritage Pima, but it's common in the Caucasian population. Maybe what you're picking up here are these are admixed individuals that have some Caucasian ancestry in addition to Pima ancestry. So they went in and they simply basically said, okay, look at the tribal registry, and we're only going to look at people that have 7 of their 8 relatives as registered as being full members of the tribe. When you do that, number 1, you see that this is a very rare site. Number 2, you see the signal entirely goes away. So this incredibly strong signal up here wasn't that GM was linked to a causal site, but rather your data consisted of hidden populations. And this was surprising because you're sampling on the Pima and the Oto reservations, self-declared individuals. They don't appear to be any structure, yet their ancestry shows there was some structure. And these individuals here, it's only a small fraction, but they have Caucasian ancestry, Caucasian admixture, maybe 1/4 or 1/8, but that's enough to give them a lower diabetes risk. So this is the problem with population structure. If you have cryptic populations you don't pick up, and those populations differ in the trait value, alleles that predict population also predict trait value, even if they're not at all linked to the trait. So a lot of work was done into how to do that, how to get around that, and most of the methods kind of use this general approach. So here's the general approach. This is basically a regression, and this here basically says I'm looking at locus marker K. I'm going to simply score it as 0, 1, 2. 0 for big A, 1, 2. So MK is a number of little a copies. So what I'm asking is there a significant effect of the trait on number of copies of this marker? That is B different from 0? That's what a GWAS looks at. These other terms here, what they basically do is they take all the marker data, and the chapter 21 goes into great detail about this, and pull out principal components of that marker data, so looking at deep structure in that data. So what this basically does is, without knowing whether or not you have any groups in there, it partitions individuals into groups. It projects ancestry on the eigenvalues, eigenvectors is what it basically does. And so this term here adjusts for the fact that this individual may have come from a particular population. So if I ignore this marker, this adjusts that individual's risk using all the other markers. This approach has been called eigenstrat, and there are other approaches like structure, etc. So the population structure is not regarded as fully solved, but it's largely solved. And the idea is you can use marker information to look and basically cluster the populations and get rid of population, and I should say correct for population structure that way. You can also correct for admixture. And the initial manifestations actually came out of plant breeding. So these human population geneticists would get these massive grants and, walk around their chest puffs out because they think they're big stuff. Their methodology they used came from animal breeding in the 80s, mixed models, and the things they used for distinguishing effects of population structure and relatives come from the plant breeders. This came out of Ed Buckler's lab. You might have heard of the program called TASSEL. And the idea is, we haven't talked much about linear or mixed models. I assume a lot of people have some familiarity. Everything in black is something you know directly, either a vector or a matrix. Everything in blue is a fixed effect to be estimated. The thing in red is a random effect. So having hidden structure can bias your GWAS. Likewise, having hidden relatives, right? So if Mike and I are brothers, well, we're going to be similar in a trait that has variation. We're also going to share alleles that aren't related to that trait. And so relatives will also give you a signal. So cryptic relatedness as well as cryptic structure will. And in fact, the way to think about it is these are both relatedness. Cryptic population structure is deep ancestry. Cryptic relatedness is more recent ancestry. And the so-called QK model, this basically adjusts for fixed effects like sex and who took the measurements and things like that. This is your actual test right here. Whoops. This is your actual test of a specific marker. And this other term here accounts for population structure. So you basically predict which population you might be and put weights on that from the markers, and you can estimate the effects of those populations. So that's estimated. And this basically then accounts for any relatives you might have. So This again uses marker information to look at that. So I talked about general pedigree methods last time. This just corrects for ancestry with general pedigree methods. So that's called the QK model. And most of human GWAS is done using some variation of that because you're correcting for population structure and recent ancestry. So that's your quick overview of GWAS. Obviously, I left out a lot of details. There's a lot of technical stuff you can really drill down in. Like, for example, if you take a marker variance covariance matrix, the leading eigenvalues account for deep ancestry Y. They're tiny features shared over almost the entire sample. The small eigenvalues, probably your recent ancestry, because only a few individuals in the population. I'm only related to 1 or 2 individuals in the population recently, but I'm related to a third of the population on a deeper scale. So all that stuff is covered in chapter 20 with examples and references. So the main idea, get a big population, look at a bunch of markers, look for marker trait associations, and then worry about cryptic ancestry and population structure, and there are ways using just markers to correct for that. So questions about the basic structure of a GWAS. And again, that's a good level to take home, is that amount of level. Yeah. Yeah, so basically to remind people about fixed and random effects, to kind of cut through a lot of the detail, one of the nice reasons for using random effects is that I can borrow information. If I'm trying to estimate my breeding value, I can use my phenotype, but if I have relatives in here, such as Mike or Nick or Lewis, I can basically use their information as well. I can borrow information across correlated characters. So for the random effects correction for ancestry, for recent ancestry makes perfect sense because what you want to do is basically you're adjusting that residual covariance structure for the fact you've got that. The reason people use fixed effects for deep ancestry is they basically pull out the first couple of principal components. And then the concern is that you're basically just trying to look for different subpopulations. And then you can estimate what the effect of that subpopulation is pretty straightforwardly. I have seen people try to use random effects for deep ancestry, but they basically just simply use, we call it a QK method because the matrix Q gives the weightings from the eigenvectors on population membership, and the shared ancestry is a covariance matrix called K or kinship. I've seen people do this just using the kinship matrix. And that does a lot of the corrections. It's not great at capturing deep ancestry, but it's certainly better than not doing it at all. So, but in general, it's been shown that using both corrections for population structure and recent ancestry does the best. But it's also trait dependent, as we'll come back and talk about. Other questions about GWAS so far? Nick?
Yeah, it's maybe a related question, but there are choices to make about how many how you go.
Sure.
How much does that affect the result?
So, great question. So how would you guys handle that? So Nick's base point was that how many PCs you put in can impact your result. How do you cut off the PCs, right? People have general rules like put in 10 or 20, or you look at a scree plot and do something like that. No. The PC1 maybe accounts for 3%. Remember, you've got the number of PCs is the number of markers. You've got 1 million PCs. So any PC, you do have a scree plot, but you've got a really, really long tail. So the way to handle something like that is to try different PC numbers and see how stable your results are. Now, there's another interesting thing, and that is PCs are based upon signals from common alleles. Common alleles are ancestral deep alleles. You can also do population structures based upon rare alleles. Rare alleles are much more recent. So if you and I share a super rare allele, we might have also shared a common environment 1 or 2 generations ago. If we share a common allele, that's probably not likely to be the case. So there are issues like that chapter 20 talks about those as well. See, the great thing is I know you're not going to read 20, so I can just say it's in there and you'll take my word for it. Yeah. Not really, because you have admixture in there as well. So in theory, you could do a PC plot and get clean separation, but think about the talk yesterday on how you do GWAS in India. That's an enormously complicated population structure. You obviously have a group that separates up, then you have this entire smear. But PCs are good at picking up that smear because, for example, you may be 80% from group 1, 10% from group 2. A PC will pick that up, whereas a PC plot per se wouldn't necessarily do that because so many individuals are admixed. There's a question over here? Yeah. Great question. Deep, deep ancestry. And the idea is that if I have a population of size 500, and suppose that you and I are siblings in there. Well, that's going to give us a big correlation. But in terms of the effect of the R1 pair out of everyone else, the signal that gets on the whole variance covariance structure is really small. Because it's a strong signal involving a super tiny subset. Conversely, if I have deep ancestry to half the room, but that deep ancestry is little tiny effects that are slightly shared, but I'm now sharing it over thousands of individuals, that will give me a big signal that the marker covariance matrix, which simply looks at pairwise covariance between individuals, wouldn't pick up. But the eigenstructure, which looks at combinations across markers, would pick up. So that's why the marker matrix, the distinction is with relatives, you're looking at pairwise corrections because you're trying to adjust the covariance structure. If you're looking for more subtle signals, you look at the eigenstructure because that signals over multiple individuals. And that's what that's the population structure corrects for. And there's some theory on that, and again, chapter 20 gives you some references for that. Another question over here? And you can read chapter 20 of the weekend and say it's not in there. That's fine, right? So what is GWAS? What has GWAS told us so far? I'm going to give you the initial things, then we'll drill down a little bit. So if I have an additive trait, that's the causal variance that's accounted for. And one thing we see in a GWAS is all the effects are small. What is the effect in a GWAS? It's how much variation in the trait is accounted for by the marker genotype. So I'm estimating what fraction of variance is accounted for by the marker. And that fraction of variance is the causal variance times R squared, which is how associated that marker is with the causal sign. So we see small effects, which means we see small variances. That doesn't mean infinitesimal, because I can have a small variance by having a modest size p and a small a, or I can have a really big A and a really small p. What we do see is there's a trend across multiple traits, I'm going to get to a bit later on, where alleles of large effect tend to be rare over many traits. So it's infinitesimal in the sense that each marker has a small variance. It does not mean the underlying alleles all have small effect. That is, the A's aren't necessarily uniformly small. It's just the case that when A is big, P is small. As you might expect from selecting, if I have an allele with a large effect, it may affect many traits, may have a lot of pleiotropy, may be deleterious. Bigger A is, the smaller its effect is. A question of concern we'll talk about a fair bit is the bulk of variation due to common alleles, 5% or more, of small effect, or rare alleles of large effect? Because we can't tell it by simply looking at these variances. We have to pick the pieces apart. So the take-home messages from the early analysis, infinitesimal-like, but now from a small variance. The other big thing is 80% of the hits were in non-coding regions. And that's conservative, at least 80% of the hits. So early on, people thought, well, gee, I'll see a hit that's right in that causes a marker that causes a nonsense substitution or a missense substitution. It'll be in a coding region. It'll be really obvious to infer. No. when you see those, they're typically really, really rare alleles. So we'll talk a lot about the bulk of the variation seems to be non-coding, and the inference there is most of the variation is probably regulatory, changes in the timing and structure. Now, a little asterisk here. A regulatory variation could be one, I've got a gene that's spliced over here, and it makes two different proteins depending upon how it's spliced. A regulatory change could influence the splicing ratios. So it would give you different products, but it would change the ratio of the products, not the products themselves. So again, we're going to flush all this out. So one of the early results he had was this, missing heritability. And the upshot was, people know from parent offspring and from full SIB, half SIB, the heritability of height is 0.7 to 0.8. You can fiddle with that a little bit, but it's still 0.7 to 0.8. Early on, people looked at their significant SNPs for height, and they could account for not 0.7 or 0.8, but 0.1. The heritability was missing, and it caused a lot of, consternation about a fundamental flaw in quantitative genetics. And as we'll see, nothing's farther from the truth. But that's the tease, the story. We're going to flesh out that story. But to do so, we have to talk about LD, right? So I'll give you an exciting mystery to kind of keep you through LD. The measure we want to use for LD is this correlation between allele I at chromosome A and allele J at locus B. So in that population, I grab out a chromosome. I want to look at the measure we're going to use is the correlation. This is the disequilibrium I talked about. That turns out, as Nick pointed out, to be a covariance. Now, there's several ways you can look at D. One way comes from Lewontin, the same Lewontin that Nick mentioned. You can have this standardized measure. So this ranges between +1 and -1, or you can have this correlation measure. Now, let's look at some real data here. Here's case 1, here's case 2. If you compute the numbers for case 1, you see D prime is 1. It's its largest possible value. We call that complete disequilibrium. What that means is, look at the data here, every time I have a big Q, it's on a big M. But it's not perfect disequilibrium. Why? Because I can have a big M that doesn't have a big Q. I've got off-diagonal elements which are non-zero. So the correlation is only a half. So even though big Q is only found on big M background, other things are also found on big M backgrounds. Conversely, let's look at over here. That's perfect disequilibrium. D prime is 0 and the correlation is 1 in that case. And so what this basically points out is that whether or not you get off-diagonal elements here are a function of the mismatch between the frequency of Q and the frequency of the marker it's usually on. If there's a mismatch, you're going to get some off-diagonal elements. And in particular, Naomi Ray basically showed you can write the correlation between M and Q is this, where basically we're making the assumption here for the strongest signal. We're saying that Q is only found on a big M chromosome. Alpha is the fraction of big M chromosomes that contain Q. This is only 1 if all big M chromosomes contain big Q. There's perfect disequilibrium. So what you see is if there's a mismatch, that is, yes, all Q are found on big M, but they're also big M without Q, then the correlation is not perfect, and only part of that signal from the causative site is transferred over to the marker variance, and the marker variance is what we score. I think an example here makes the point. So here I have N for Nancy and M for Mary. I say that because I'm not good at enunciating N and M. So what I have here is in the population, 60% contain big M and 5% contain little m, and then big Q is only found at 3%. So I go ahead and work that out. What I find is that in both cases, D prime equals 1, complete disequilibrium, but I don't have perfect disequilibrium. So what you might expect is big M is much closer to Q than big N is. But let's look at the R squared. When you work out those R squared calculations, what you see is the R squared between big M and big Q, the closest sites is 2%. The R squared between N and Q is 0.6%. So this one is right next door, but it gets a smaller R squared. Why? Allele frequency mismatch, right? That's 5%, that's 3%. The allele frequency mismatch, N and Q are much closer in how their allele frequencies change or co-occur. And so if I have a common allele, big M is a common allele, it doesn't capture this rare allele well. Only 2%, R squared. So only 2% of the variation from big Q is captured by big M. Conversely, for the rarer marker here, if it's in disequilibrium, it captures more of that signal. This marker that's further apart captures about 60% of the causal variation. So a big misconception in a GWAS is the highest R squared is the one that's closest to the site. Doesn't have to be the case at all. And mostly that arises from these mismatches between the frequency of the causal site and the frequency of the marker you're looking at. Question? Yes? Hang on, let me get back up here. This one here? Uh-huh. So if you look over here, yeah, that's a typo, good catch. Yeah, I flipped these around. Yep, thank you, I'll change that in here. So this should be 0.6 and that should be 0.3. Yeah, very good catch. Mike, you missed that. Oh, you actually haven't reviewed this chapter yet, so.
I had a different question.
Oh, yeah.
Isn't there a property of R squared that the maximum value is only 1 if the frequencies of sites are equal?
Yes, that's what it basically shows here. Yeah, that's exactly right. But as that mismatch becomes larger and larger and larger, R squared gets really, really tiny. And the tiny R squared means you're only capturing a small fraction of the causal site through the marker variances. Yeah, I think I flipped these two. No, I flipped these two. So if you plug in N here, yeah, I think I flipped these. If you were actually work it out here, so let's work out the math here, right? This is a much bigger difference than that. So this is larger on top, gives you the 0.5. So yeah, I messed up I flipped the answers here, but you still have that big little n gives a stronger signal than big M. So since you're excited about it, go ahead and work those out and you'll put the right numbers there and you'll see that. So this is the result there. Let me get back to you on that. Yeah, because the math is correct. I probably didn't type it down correctly, so we'll check that out. But you do in fact get this result here that and so think about the equation up here, right? So what is A sub n? That's what fraction of big M contains big Q, and the fraction of big M that contains big Q is 0.6 minus 0.3. It's only sorry, it's the other way around. Only a tiny fraction of big M contains big Q, whereas a large fraction of big N contains big Q. Yeah, we'll talk about it. I'll fix that. So the net result of these differences and mismatches is if the causal and marker sites mismatch, then LD between them will be small no matter how close they are, even if they're in what's called complete disequilibrium. You only find Q on one marker background. What we score is the marker variation, and what that is R squared times the causal variation. That's what we want, but if I have a rare allele and I'm using common SNPs, R squared is going to be small. I only capture a small part of that. So the marker SNP with the strongest signal is likely not to be the closest SNP to the causal site. And we can go a step further. And what a number of authors have done is they've said, okay, I'll do a simulation. I'll know what the causal SNP is. I'll run a GWAS, and I'll use whole genome sequencing. So whole genome sequencing. So what they're doing is they're looking at an allele. It's a causal allele at high frequency, 50%, has a large effect. Human geneticists often work in odds ratios because looking at diseases. This gives you a 1.5% increase in the risk relative to a random individual. That's a big effect. Only 80% of the time will the lead SNP correspond to the causal SNP. That's with whole genome sequencing, a large effect of an allele, and also a high frequency. That number goes from 80% to 3% when that allele is rare, only 5%, and it has a small effect. The odds ratio is 0.11, so only a 10% increase in your risk. More direct, another simulation, if the causal SNP is common, then under a whole genome sequence, 80% of the lead SNP with the highest p-value, is within 10 kb of the causal SNP. So 80% of the time it's within 10 kb. It's not bad, but it's not sitting on it, 10 kb. If I do imputation, so this is whole genome sequencing, imputation means I look at 1 million SNPs and I basically predict what the other missing SNPs are. With imputation, that goes down to 25 to 35 kb. So there's not a big increase with whole genome sequencing. Yes, it's an increase, but it's not, is it worth a whole genome sequence to go from imputation to that? You get a narrow interval, but it's not 1 kb, it's a little bit. Conversely, if I have a rare SNP, under whole genome sequencing, 95% of the lead SNPs are within 5 kb. If I take this same scenario and use imputation, that is, guess these from a reference set, it's now only 37% are within 5 kb. So bottom line is, whole genome sequencing is not that helpful for common SNPs, but it can be useful for rare causal SNPs. But of course, you don't know that because you're simply looking at a marker effect. It's error, but it's also the fact that these things are highly correlated because you have very high LD, and so a little bit of noise can flip that up. And so so it's a combination of those two. It's the fact that you have high multicollinearity, and so, for example, you might have a certain marker that is only on one chromosome, not with your causal site, but on that one individual that by chance they have a value that's 5% larger. And that could kick you over the top. So the bottom line is, people often thought that if I do whole genome sequencing, I will score the causal site. Yes, you will. I will pick out the causal site. You may not. You're going to be close. And chapter 21 talks about different approaches to try to find and map where those causal sites are. Not necessarily, because that top SNP could be a lot further away but have a closer allele frequency match than the SNP below that. So the bottom line is that the GWAS gets you close, but it doesn't get you the SNP. But it gets you close. And whole genome sequencing won't close that difference. So there are other ways to do that. So the mythical thing people are trying to get is what's called a QTN. Not a locus, but a quantitative trait nucleotide. This signaling nucleotide impacts the trait of interest. Yeah, so basically if I have marker information, so one thing is I could have a hybridization chip, where basically I take the DNA and I can score the SNPs at 100,000, 1 million, 2 million randomly chosen sites, and that is called the less than whole representation genome, or I could have a whole genome sequence where I know every single step. So most of the GWAS is done by using SNP chips, because they capture most of the genome. If you do whole genome sequence, you'll capture everything, assuming let's not worry about errors, but you won't really gain that much for common alleles. You go from a uncertainty of about 25 kilobases to uncertainty of about 10 kilobases. That's good, but that effort may be better spent in other ways of looking at things that we'll talk about.
Yeah, I guess what I'm wondering about is the initial investment in the chip versus whole genome sequencing.
Well, yeah, and so Mike's question is the initial investment in chip versus whole genome sequencing. So yeah, with non-model organisms, just sequence, right? Basically, a chip is the flip phone error, and whole genome sequencing is your cell phone. Technology has gotten cheaper and cheaper and cheaper and cheaper. Again, as Rebecca Dorsch says, never marry a technology, it will leave you. And so people use SNP chips now because they've got them developed for certain species like humans, and they're quick to use and cheap to use. But if you have a model organism, you're probably a new organism, you're probably better off getting into whole genome sequencing or genotyping by sequencing other approaches. You develop a chip if you're going to be sampling again and again and again. But my guess is by the time you develop that chip, genome sequencing is just dirt cheap. I mean, a human genome now is $60. You can argue about the coverage, but yeah, so there. Let me give you another cautionary tale. So FTO is a gene that's well known in metabolism, and in a GWAS in humans, they found 90 variants that were in strong LD with each other that mapped in about a 50 kb region that spanned introns 1 and 2 in this gene. Strong signals, highly reproducible. This is for body mass index. And what they found is individuals that are homozygote for risk alleles averaged about 3 kilograms higher than individuals that were homozygotes for non-risk alleles. If you go into mice and knock out FTO, you get leaner mice. Man, you couldn't have a better system, right? It shows you that. But then you look at it more carefully. And it turns out that these sites within the introns are almost certainly regulatory. There's no structural change here. The FTO protein structure is the same. If you look at those regulatory changes, they're highly in risk for cis-acting factors, factors that bind on the chromosome and influence adjacent things, like transcription factors and enhancers and PNS1 cypertensive. However, none of these mapped as EQTLs for FTO expression. Remember, I'll talk about this a bit more. We can take the level of RNA at a specific gene and treat that as a quantitative trait. It shows variation. We can ask, are there loci that influence levels of expression? Well, these sites within FTO, none of them were EQTLs for FTO expression. None of these sites in the intron of that gene influences expression. However, 11 of these influenced this nearby gene called RIX3. Why? DNA is not a rod, it's a rope. And the DNA looped around such that this gene, which was several kilobases away, now interacts with those cis control sites, which are in this region. These are genes which are expressed in the brain. When they're expressed, and they looked at those which were expressed, had eSNPs for this gene expressed in the brain. When you do that, they're associated with BMI. So what you're having here is these hits in the FTO gene are genes that influence expression of another gene in the brain. It was that signal in the brain that was impacting body mass influence, not FTO. It just shows you how complicated these things are. And the reason people drill down in this is if you get a gene that reduces body fat, you have a yacht. And you actually have a fleet of yachts. So there's a lot of inference to try to get that straight. So now let's go back to the case of the missing heritability. Notice here the little burglar sneaking out the back with the heritability. This is a paper that caused a lot of angst. And here's my favorite quote from an evolutionary quantitative geneticist, Ben Shelton. This is a little bit of a kind of, an attitude quote. So again, let me remind you what the missing heritability was. There are all these classical methods that showed the heritability for height would be 0.7 to 0.8. Reproducible, classic, little variation with that. When they then went in and took they took SNPs that are found by GWAS. So they spent $500 million to put the infrastructure in, and then found SNPs and put the significant SNPs into a regression. They could account for about 3% of the heritability. So Ben had this great little quote, there's something remarkable and encouraging about the fact that a centuries-old method, parent-offspring regression, requiring no more than a ruler, a pencil, and I suppose a slide ruler, outperformed by an order of magnitude the fruits of the genomic revolution. So that was kind of the evolutionary quantitative genetics flexing on the human people. So why is this the case? I think you've got an idea in terms of R squared, right? If my R squared is only 0.1 for a given marker, I'm only going to pick up 10% of the causal variance. So here's the idea. What you did is every marker above the significant threshold, you put that into your regression. Well, number one, you've got all these markers down here which don't rise above that really stringent Bonferroni correction threshold, but these are probably biologically relevant, so you're missing all these. Number two, even if you put these in, they only pick up R squared of the actual causative variation. If there's a big mismatch between the marker frequency and the causal frequency, R squared is going to be small. So it's not at all surprising you had missing heritability. That's kind of points out that. And so as population size got bigger, you started capturing more. So in 2008, an initial GWAS of 40,000, they had 27 significance, they captured 6%. The percent here is of the actual heritability. So this would be 4.2%, so 6% of 0.7. You go to 180,000, you have 200 hits, you now recovered 14%. You go to 500,000, you now have 700 hits, you capture 20%. So a lot of this comes out of Peter Vischer's group, so and as part of a consortium. So this is 700 hits. Suppose you take the top 2,000, 3,700, 9,500. Now you go from 20 to 26%, 30%, and 36%. 2018, 700,000. You take the top 3,000 hits, you get 37%, or 35%. There are ways to use all the markers at once. These are regression shrinkage methods. And when you use all the markers at once, 4,000 individuals, and again, the idea here is you put all the markers in the regression, and you've got methods that basically shrink the signals from those which aren't very important. This is a Remel method. So you can now account for 62%, take up to 44,000, account for 80%. You got interesting things. For example, this is all height, but for schizophrenia, what you can show for height in schizophrenia, if you look at markers on a chromosome and compute a heritability just for that chromosome, longer chromosomes had higher heritability, so as you might expect if markers were uniformly scattered across. For schizophrenia, they showed that 70% of all 1 megabases region contained at least a risk site. So it's all over the place for schizophrenia. Likewise, for height, larger chromosomes have higher heritability. So height is super polygenic, it's scattered all over the place. 2022, 25,000 whole genome sequencing. If you use just the common markers, you could account for 68%. If you imputed, so you ignore the whole genome sequencing, predicting rare alleles, you count for 50%. If you use all the data, you now account for 90%. So using all the data allows you to pick up the rare alleles. Also, you've got sites which are sitting right on top, so R squared is now 1. Current estimates on height are between 95,000 and 100,000 causal sites. That's not loci, that's sites. So it could have a locus with 15 different sites that show up at different times. Jean-Luc Loic at UQ did a lot of this work, and what he showed was it's concentrated in about a quarter of the genome. It's about a quarter of the genome where you tend to get multiple sites which impact height. But there's a huge amount of sites. It's certainly super polygenic. So far, one of the limitations of GWAS, it's going away as genome size, as samples get larger and larger and larger. If you don't have the causal site in your study, then it's tagged poorly by a common allele if it's rare. If you go to a whole genome sequence, it now is in your study, and it's now obviously captured with much higher precision. So a lot of the signal is coming from A, rare alleles, but B, it's also coming from common alleles of small effect, right? So you get signal. And I think the current estimate is over 2/3 of the signal for height is coming from common alleles, and 1/3 or less is coming from really rare alleles. So questions so far? So what's generating all this variation? This kind of reminds you about the exciting route from DNA to your trait value. So for example, if you have histones, you've got methyl groups and acetyl groups, and those influence whether or not the chromatin is open or not. So obviously, these have genetic variation. You can basically ask, are there sites that influence whether or not a site is methylated or acetylated? And you find those. Some of those map, as you might expect, very close. Some of those map on different chromosomes. Likewise, you have all these features. When you decide to turn on a gene, you've got promoter binding, you've got messenger RNA making, you can have different splicing. Polyadenylation levels refers to messenger stability. You can have different amounts of polyadenylation. Then you can have different rates of translation. So each of these intermediate steps has genetic variation, and those are steps where you can have little effects that might influence the final downstream product. You can go even further, but basically says the thing. Then you take all these things from gene and run it through a very complicated biological network. So these are now protein-protein interactions. So there's huge amounts of areas where regulation can impact your final trait. And the idea that many people have, and we'll flesh this out a little bit later on, is super rare alleles tend to be large effect mutations that impact amino acid sequencing. Not always. So they're super rare because selection knocks them down, but their effects are big. But most of the variation is caused by small effect changes influencing all these different regulatory steps at different sites. So now it's not perhaps surprising that there are 100,000 different sites impacting height. Suppose that that a locus for height is impacted by an RNA that binds to it and helps facilitate splicing or helps open up the chromatin. Now on this chromosome over here, I have a random DNA sequence. It now has a mutation. That mutation now gives it a very weak binding affinity to this RNA over here. So a little tiny bit of that RNA now binds to this site, totally non-functional. That will change the regulation in a really tiny way. And so this non-functional site over here is impacting regulation. And that little change in regulation will impact into a little change in trait value. So most of the quantitative variation seems to be variation in regulation. That brings us up into the whole idea about regulatory SNPs. So here are basically all these are features we can measure. Is this site methylated? Well, it's 0, 1. I treat that like a disease gene. I can map sites that impact that. I can map SNPs for that. Just like I can map SNPs for height, I can map SNPs for expression of FTO or expression of any of the 30,000 genes in the human genome. Or I can look for protein levels or chromatin assembly or chromatin acetylation or histone acetylation. Those are all things I can look for variation, and you find them. So these are regulatory SNPs, and the idea is these regulatory SNPs are some of those unknown regulatory factors that influence your trait downstream. The early example of this was actually for protein spots. This was actually done in QTL. A group out of France did random protein spots in maize and looked at spot volume. That is, these were proteins that didn't know what they were, but spot volume is production. Then it went on to the transcriptome. So people, first of all, did microarrays and then got technology such as RNA-seq that allows you to go into a cell and count up how many copies of each individual transcript there are there. So what I can do is I can go to my transcript Y here, score that, and then do a GWAS, look at how transcript Y varies over individuals, do a GWAS and see where I get hits. So for example, I might get something like this. So on here, This is you can flip this axis either way. This is the transcript I'm looking at. So it says for this transcript right here at this genomic location, there's a site that influences this transcript. For this transcript over here at this site, there's a site that's located away, and there's a site that's basically right next to it. This line is on that. And people have historically used cis versus trans. Remember from your old days of prokaryotic genetics, cis means a binding site on a DNA molecule that directly impacts an adjacent site. It only impacts stuff that's on the molecule it's bound to. Trans is a diffusible factor. Well, people started calling things which were close cis and far away trans, when in fact doesn't have to be the case. Something far away could loop back. So now they use the term local and distal. So if you do that, what you find is something like this. So for example, what this says is that this location here influences all these transcript levels. We call that a hub. You see there's a lot of things which have cis EQTLs, and for example, this site over here is affected by a trans, is affected by a distant, and a second distant one. So you can do these, and they're called waterfall diagrams. They look much more complicated than this. But this is a way of looking at gene variation in levels of transcripts. You do the exact same thing for splicing ratios or for protein production. And so this is what people are now doing, which is why I call it quantitative genomics, is they're moving off a trait and they're using these high-dimensional genomic features such as the transcriptome, the proteome, the metabolome, the methylome, et cetera. So 21 talks about some of the problems of basically finding cis versus trans, but a model that's really helped people is the idea about mediation. Suppose I have a SNP I, and I can show that SNP I impacts the transcript from gene J, and I is quite a bit away. So we would call then, this would be an eSNP for trait J, and it would be a trans or a distant eSNP. These are obviously hard to find because there's a whole bunch of stuff you have to search through the entire genome. One of the breakthroughs that came is that people thought, if you have what's called mediation? So SNP I is a cis EQTL that affects the transcript of gene it's adjacent to, and that transcript then affects gene J, and that affects the transcript of J. So instead of looking for trans, the idea is you search among those that show eSNPs and use those then to search for trans. You can also break these down a little bit further. You can ask about the different pathways. So for example, here I have a SNP, and this could be a transcript, or it could be the trait value. So I can basically ask this is how that SNP affects the transcript of I, and this is how I affect the transcripts of J, and this is a pathway that's independent of T that goes directly from SNP I to SNP J. So these kind of path analysis talks, this is called the mediator. I've got an intermediate. If this path was 0, if I knew this value, I could completely predict that value. So people can go ahead and do this and then ask what are the impacts of these different mediations. Just because time is short, you can basically make several regressions. So this is regressing the transcript level of trait J on the SNP count of SNP I. Beta 1 is that impact. Then you can look at SNP I on its transcript I on transcript J, and then put those both together in a joint regression here. So this would basically, if the only effect of I is on J, this term would be 0. This allows you to separate out I into J and then I into T, and this ratio here is simply the amount of mediation that's accounted for. So this is kind of quickly, this is sort of the methodology that people are using to try to look at some of the impact and input of some of these intermediate values up there. When they do that, they see that mediation is common, but it's not complete mediation. That is, that there is an effect from here, but there's also a secondary effect from there. So there are other pathways that things are going through. There's also, when you come down to localization, one of the approaches people try to use, I have a series of SNPs which are all high. Well, one way to think about what's a good SNP is maybe that SNP impacts the trait and also transcript levels impact that trait, and that SNP seems to be an eSNP for that transcript, co-localization. So under a cis mediation model, This is the chromosome. That box is my SNP. That box impacts the transcript, and the transcript levels what impacts Z. Or I could have linkage. I could have two tightly linked SNPs, one that impacts the transcript, one that impacts Z. I'd still get a TZ correlation. Or I could have pleiotropy. I impact both the transcript and Z without there being a connection, or of course I could have all three. And those path methods I showed you before, which are discussed in 21 and Appendix 2, tell you how you can kind of decouple those. So the take-home message from here is that now people are trying to get a handle on what is some of the regulatory variation that seems to underlie most of trait variation. And the reason that's relevant to here is those are obvious targets for polygenic adaptation. Regulation at sites you might not think would be impactful, that impact transcripts, that impact the trait level, or impact splicing, or impact protein stability. So, questions about this idea about mediation? You have an intermediary, a SNP, impacts one, which impacts the second one. We now have ways, if you can measure SNPs and traits, that we can kind of work those out a little bit. They're basically partial regression methods, and chapter 21 talks about them. The key idea, though, is that, number one, most GWAS is mapped to non-coding regions. And the idea is that most of those non-coding regions probably are involved in regulation. Either eSNPs directly impacting something, or a trans-SNP may be acting through some intermediate to do that. So a lot of the variation isn't in differences in protein coding sequences, it's the differences in amount. And the idea is that rare SNPs are very often large effect SNPs, and they're very often things that involve disruption of protein coding. Common SNPs have small effects, and they don't map to coding sites. There are probably some of these regulatory steps that people are catching up. So questions about that so far? Yeah, so great question about, so if you're going to do these studies, one of the things you have to ask is, think about expression. What tissue is expression relevant in? Right? It's an ascertainment issue. So, for example, you're working on a Arabidopsis in nature, and you're looking at transcript levels and trying to predict seed production. And you're focusing on the meristematic or the seed tissue. What if all your action is in the roots, and it's transcript levels in the roots that don't appear in the seed tissue that are giving your differences in there? So when these functional genomics things are really nice, but they require you have a reference cell type or developmental stage or developmental time that you care about. And so there's obviously ascertainment bias in there. Often these are done by whole organisms, so you grind up the fly. Now people are trying to get a little bit more precise. But even with something like yeast, right, you want to have them in the same developmental cycle. If you're in stationary phase, your transcripts are rather different than you're in growth phase. And so there is an ascertainment in terms of not looking at the right tissues. And what those right tissues are, we don't know. And so the literature now recommends what they call agnostic screening, where the methods where you can basically test all these things over all the different tissues and do a p-value correction and come out of it that way. So I mean, this stuff is really powerful, but there's also a huge amount of infrastructure that's needed to get them right. And again, if you guess the wrong tissue, you may have a strong signal you entirely miss. And so you can go, well, I know this gene is involved, I just haven't picked the right tissue yet. You can get stuck in that as well. You have to use what the data tells you. Let me finish up then by the common versus rare alleles. So generally speaking, under drift, a common allele is an old allele. Now under selection, if it's selected for, a common allele could be young, and a lot of tests for ongoing sweeps are based upon the fact that alleles show features that they shouldn't have because they look young relative to their frequency. But under weak selection, typically a common allele is an old allele. Are most of these regulatory, potentially? Rare alleles are more recent. I mean, there are exceptions, but the idea is that if you're a rare allele, you either have just arisen, And if you're deleterious, you're not going anywhere, so you're not going to spread much. And a lot of these could be mostly structural. So most hits have small variances, but they could be common alleles of small effect or rare alleles of large effect. And again, there's this common pattern of this inverse correlation between effect size and frequency, such that over a large number of traits, GWAS hits with large effects tend to also have very low frequencies. So how can we adjust for that? Well, one way to think about that is imagine that the effect size is independent of the frequency. If it's independent of the frequency, well, this term here, 2a squared x1 minus, that's the causal variance. What's the frequency? Well, if you assume neutrality in an equilibrium population, you've got the Watterson distribution. So the frequency is 1 over x times 1 minus x. If you do that, you then get a constant. And what this says is the fraction of variations accounted for by alleles at frequency less than 12% is 12% of all the variation. You have a uniform here. So if you do this calculation, rare alleles, for example, 1%, could only account for 1% of the variation. Now that's under the assumption that the allelic effect is independent of the frequency. The frequency comes in here because that's basically the variance, but that allele effect, larger effect alleles could be smaller, so the frequency may tell you something about the allelic effect, and several people have made models for that. So for example, here is one particular model. It says, what's the allelic effect given the frequency? Well, there's some value where it has no effect, and that 1 minus pi 0 is called the polygenicity, the fraction of SNPs that could have an effect. Then when you draw effect, you get this scaling here. So what this scaling says is, given the frequency, then I draw from that, and this variance gets larger as as this term gets here. And then you can go and basically then estimate that. If you do that, you can then ask what fraction of variation is accounted for when the minor allele frequency is x. You get this term here, you get the 1 plus s in there. You can then estimate that from using maximum likelihood. It's kind of sort of thing, this is kind of a different way what Peter Keatley is looking at stuff. When you do that in Biobank, you get S values ranging from minus 0.6. Negative number means you get a larger effect when you're smaller. An S of 0 means you have no effect. Your allelic effect is independent of the frequency. And so what you basically see is that most of the S values are negative. So for most traits, alleles of large effect are rarer. And likewise, the polygenicities had a median value of about 5%. So about 5% of all SNPs can potentially, under this approximation, can generate variation, and it ranges from less than 1% to about 14%. So you can do a bunch of tricks like that, and chapter 21 talks about this. Yeah, 2 questions. So on this parameter, what? Oh yeah, so there are lots of ways to do that. I like a little better term. So it's a better term involving the kurtosis, that chapter 21 talks about this. But in these initial experiments, so this is kind of the structure that a lot of models use for genomic selection, in that if you take a random SNP, there's some SNPs that no matter what they're not going to mutate and impact your trait. So they have no effect on the trait there. It's a point mass at 0. Pi 0 is a fraction of SNPs that you estimate from here have no effect on your trait. So 1 minus that is the fraction of SNPs which could have an effect. If they have an effect, then that effect is drawn, and it has an average effect of 0, because it could be positive or negative. And then the spread about that effect, as that spread gets bigger, you get more extreme effects. And that's kind of how these models are built. There's several different ways to build models like this, and chapter 21 talks about several different approaches people have used. There's this kind of disconnect between the human population genetics, which has gotten, since 2015, interested in looking at signature selection genome-wide, and the kind of classical stuff that Nick and Michael and people like that did. Adam Airwalker had a paper that kind of split the difference that the human people cite, but the human people don't cite all the other classic stuff. But they have really interesting data. Question? Sorry, really speak up. My hearing's bad. I captured about half. Let me paraphrase it. So you're asking about biologically why rare equals protein and common equals regulatory. And the idea would be that if I'm going to put a random mutation down, where is it more likely to have a big effect? If it disrupts a protein. I mean, generally speaking, right? Versus throwing a random mutation into a regulatory network. Certainly some can have a big effect, no question about that. But as a whole, if I throw down a random mutation in the genome, and I tell you that I hit a protein coding sequences, your expectations is probably going to have an effect. If I throw it and hit somewhere else in the genome that's not a coding sequence, your expectations, it's probably not going to have that, on average, that big of an effect. And the idea is that and the data show this to some extent, that rare alleles are often in protein coding regions. Common alleles are rarely in protein coding regions. So the idea is those rare alleles that disrupt a protein coding sequence have a large effect, and therefore selection is removing them, and therefore they never get very high. That's kind of the logic on that. Let's put all that stuff together in the last minute or so. So Jonathan Pritchard and colleagues came up with this idea called the omnigenic, all-seeing model. And there's a lot of controversy, which I think is kind of weak controversy, in that all that Pritchard and his group did was take stuff that everybody's talked about and package it up nicely. So I think some of the controversy is that, you know, they get more credit than some people think they might, but there are also some other issues. So the idea is you've got a bunch of genes here called core genes, and those core genes transcript impact your trait. Then you've got a boatload of genes up here called proliferal genes, which have weak interactions to the core. So the idea is and this is because the network structure of complex biological systems, all these proliferal genes have really weak effects that impact expression in the core. Any one has a weak effect, but together effects could be rather dramatic. So this could be most of your common alleles. The core genes, if you knock out a core gene, those could be your rare alleles that have really, really big effects. So one of the controversies is that what Pritchard said is you should focus on rare alleles, because rare alleles are probably those that knock out the core genes, and the core genes may tell you the most about the biology of the system. Other people have said, just get larger sample sizes, because larger sample sizes will allow you to pick out the effects in these proliferal genes as well as the effects in these core genes. So that kind of leaves you where we are right now. The big take-home lessons are, it's an old idea. Wilson really harped on it. Mike mentioned Alan Wilson harped on it, which was that a lot of variation we see is regulatory. I think the surprising thing is probably a lot more than most people would have thought. Most GWAS hits are regulatory changes. And again, I want to stress, GWAS looks at segregating variation. QTL mapping looks at fixed differences between populations. So you take your segregating variation and you filter it through selection. So line cross results from GWAS may be more relevant to adaptation, but they're a very biased picture of what standing variation looks like. For standing variation, most variation for a trait seems to be small regulatory changes, which cumulatively over a very large number of sites can have a big effect. You likewise can have large changes, which are very rare, often but not always. There are examples which aren't protein coding, probably major regulatory changes, often in these protein coding regions. And one way to think about this is your trait is embedded in a network. And the network may have a couple of core constituents, maybe a couple of hundred, whose expression has a strong impact on the trait. But the expression of those core genes can be modified by thousands of other proliferal sites, because biological systems have all these interconnections in there. So that's kind of the current view, subject to change next week, about what generates quantitative variation. And the bottom line is standing variation in well-studied systems. Humans may be unusual, but there's a lot of data on them. Maize is well-studied as well. It's a bit unusual as well, but in maize as well, most GWAS hits in almost all organs which have been looked at, go to non-coding regions. Those few examples that go to coding regions are where people specifically are looking for certain traits, like for example, coat color. Coat color, maps to obvious structural genes and then some regulatory genes as well. That's kind of now where we stand. So what I'll do next week is I'll give kind of a methods lecture where I'll kind of talk about different methods for multiple comparisons and also combining p-values. We'll talk about meta-analysis, then we'll talk about how to handle large parameter problems, things like model selection and penalized regressions, and just a quick introduction to some basic methodology if you're looking at a lot of data that are things that might be useful for you. Okay. So other questions before lunch? Great question. So there's a difference between the common allele as a marker and the rare allele as a causal site. So if I have a common allele, it's not going to tag. That is, the R squared with a rare causal site will be really small. So it'll be hard to pick. So it's a double whammy. If I have a rare allele of large effect, number one, it has a small variance to begin with. Number two, the effect you see is R squared times that marker variance. If R squared comes from a common marker, you're not going to see it, because R squared between a common marker and a rare allele, that R squared is very small. If I look at the causal sites themselves, the expectation is that a lot of these regulatory changes probably have minimal or weak selection, maybe in the drift barrier that Mike talked about. So because of that, they'll behave roughly neutrally, and so some of them will drift up to moderate frequency. So common alleles are old alleles. They're probably old because they probably haven't been under that much negative selection. Now, there could be rare cases where they're favored and they'll be common, but those are going to be unlikely. Rare alleles, rare causal alleles, are new alleles, recent alleles, and those are probably alleles which are deleterious that are on their way out by selection. There's certainly some which are not, but you're also going to get a pool of rare alleles which are deleterious, and maybe you're a new mutation, maybe you've only been around a couple of generations, and they're never going to get very high in frequency. But their effect sizes are big. So in theory, you could select for them if their advantage in your selection scheme outweighs their pleiotropic disadvantage. And so one interesting idea is a lot of adaptation might come from that pool of rare alleles. The problem is, if they're too rare, it's hard to reach down and grab them because they'll be lost by drift before selection can pull them up. Right, so when you run a GWAS, all you score, unless you hold genome sequence, you have markers that you're scoring. Yeah, and so when you run those common markers, you only pick, I'm not quite getting your question, you only pick up, the chance, well, so the answer is it depends, because it depends upon the allele frequency mismatch. But I guess the more relevant question is that your lead SNP is almost never near your causative SNP. It's within, as I showed the simulations, within 10 to 20 kb. In that, for a variety of reasons. With whole genome sequencing, that number gets a little bit lower, but if your causative site is common, you map with less precision than if your causative site is rare. And the reason for the rare causative site is you're going to pick up all the signal in whole genome sequencing, whereas you would only indirectly tag that signal with a more common allele if you use a chip. We can talk more. A question over here first, I think? No, you didn't? Okay, so lunch comment. So you want to wait till after? Okay. Oh yeah, you can build reverse arrows in your path diagrams. That's easy to handle. You can put it in there. The model, your dimensionality gets bigger, but with enough data you can test it.
I suspect that
well, yeah, the buffering through system. Yep. And the feedback I'm getting is lunch. So we have plenty of time for discussion, and I will correct that one problem. I made a typo, I'll fix that. Okay, so great. We have Daniel is on after lunch, and I think this afternoon after that, and then we're free.